We have already undergone a few lessons where we have discussed the introduction to transcription, the prokaryotic transcription process, and its different phases, including the initiation, elongation, and termination stages. Also, we have some idea about the particularly post-transcriptional processing. Now, a different topic, that is transcription in eukaryotes. Before discussing in this topic, let me make it very clear that transcription in eukaryotes is a very interesting topic, but at the same time, it is a difficult topic, and a lot of new things are discovered each day, and the plethora of information is gathering in every new edition of a textbook. But naturally, this topic for the undergraduate examination is not very important. A detailed knowledge is not required. There are certain things which we shall discuss, which are most important for examination, and rest of the thing we shall just have a bird's eye view. So with this introduction, let us see what is transcription in eukaryotes. The multicellular eukaryotes use differential transcriptional regulation to create different cell types. So that means, what does it mean? Means that the proteins which are formed in brain are not the same protein which are formed in liver. Although both the brain and liver cells have the genome which can synthesize both the type of protein. So there comes the differential transcriptional regulation. So differential transcriptional regulation. So that is a very interesting thing. Next, the most striking differences from prokaryotic transcription. See, we have just mentioned some points. Number one point is nuclear membrane. That does mean that in prokaryote there is no nuclear membrane. The chromosomal material, the chromosomal element is present in the cytosol itself. So, when the transcription process is undergoing, the RNA transcript is formed, as we have said, that this RNA is formed. Some part of the RNA which is now come out from the RNA DNA hybrid will be translated. So translated, and the other part there is transcription is going on. So while the vehicle is moving, some passengers are coming out, and they have started their marketing like that actually. So this is known as co co-translation. Co-translation. The term we use is co-translation. Co-translation. So transcription in prokaryote is co-translational. So it is neither separated in space, neither separated in time from translation. But in eukaryotes, the scenario is totally different. The translation take place in the cytoplasm, and transcription take place in the nucleus. So first the RNA have to be formed, then it will be migrated to the cytoplasm where the ribosome will take care of the translation. So this is one thing. Next, complex regulation, transcriptional regulation. So in case of the eukaryote, the transcription machinery include the polymerase, the DNA, and the DNA means the transcribed element and the promoter site, and these are the basic things. So the basic machinery is there, but in addition to the basic transcriptional machinery, including what I have said, polymerase, DNA, and promoter site. There are regulatory elements which are situated much away, much distant. Like we have our regulatory authority, that is MCI, which is in Delhi. So, so the regulatory elements are there in the chromosome, but they are much distantly located, and they might directly or indirectly. How indirectly? Because they may interact with some proteins, which those proteins which may interact with the basic transcriptional machinery, that is the promoter and polymerase and all, or they may synthesize some protein which may come into action like that. So those regulatory elements are the regulatory DNA and other proteins. They actually regulate the basic transcriptional machinery. Basic transcriptional machinery is something which can synthesize the DNA, but it is all regulation, and it is up to a basic level. But total regulation, whether it will be augmented, whether it will be suppressed, is governed by some other genes or DNA situated far distant from the gene. So that is why it is a complex regulation process. Finally, it should be number three, which we have wrongly showing here number one. It is number three actually. So number three is RNA processing, which we have already done the class on post transcriptional modification. We must remember that the prokaryotic RNA is mRNA is directly from DNA. Prokaryotic mRNA is directly formed, but in case of the eukaryote, it is first hnRNA. So DNA to hnRNA. hnRNA is known as the primary transcript. Heterogeneous nuclear RNA. It is known as the primary transcript, and the primary transcript thereafter is converted to mRNA, and the process is known as post-transcriptional modification. So there are some other aspects of post-transcriptional modification, but primarily it is in case of the eukaryotes that it take place. And also in eukaryotes, usually one gene has one gene is like that one gene, but there may be exon and introns, as we have after the synthesis, the RNA also contain the exon and introns. Genes and intervening sequences, these are the intervening sequences, and RNA will contain the exon and introns, as we have seen. In case of the prokaryote, there is no question of any exon or intron. In the RNA or in the DNA itself, there is no such thing. Rather, in case of the prokaryotes, the DNA are gene are situated side by side. That is why they are known as polycistronic mRNA. The RNA is that form is a polycistronic mRNA. And many actually such genes are actually many such proteins are formed from same gene, adjacent genes actually. Three different kind of RNA polymerase depending upon the template specificity and location. So in case of the prokaryote, we have mentioned only one polymerase with 5 subunits and a huge enzyme. But in case of the eukaryote, several different polymerases, and they have got different subunits. They're all large proteins. So the point to note is that they are all large proteins containing 8 to 14 subunits and having molecular masses greater than 500 kilodalton. So they are all large protein, and we will be concerned with the RNA polymerase type 2 mostly. But let us first have a look at all of them. Polymerase 1 is located inside nucleolus. It transcribes a tandem array. So it transcribes a tandem array of genes. What is tandem array? Tandem array means several genes which are situated closely but with a distance. And in case of this Pol 1, it actually transcribes the 18S, 5.8S, and 28S ribosomal RNA. These three are the ribosomal RNA. They are tandem array, means they are placed one after another in the DNA, and the same polymerase actually transcribes all of them. Only one ribosomal RNA that is left out, that is the 5S. So this is the 5S. This, along with the transfer RNA, they are actually transcribed from another set of genes, and they are transcribed by RNA pol 2, 3. So it is the RNA pol 3 that take care of these two types, that is the 5S ribosomal RNA and transfer RNA. And RNA pol 3 is located in nucleoplasm. So only nucleolus that contain the RNA pol 1. But our discussion, we are major concerned with the RNA pol 2, because that is the enzyme that transcribes the mRNA, which we are mostly concerned, and that is located in the nucleoplasm. Also, in addition to mRNA, the byproduct actually of the RNA pol 2 transcript, they have got several small RNAs, and that includes snRNA, which is involved in splicing, and other small regulatory RNA like miRNA, siRNA. So RNA polymerase 2 is very, very important, because the mRNA is something which is a precursor of all the proteins we know, and also the small RNA like miRNA, they are the important for the regulation and splicing snRNA, that is the necessary for the ultimate formation of the mRNA. So RNA pol 2 is very important, and we are going to limit our discussion mostly to the RNA pol 2 only. Now let us see some of the effect of a poison, that is the amanitin. Amanitin, alpha amanitin, is produced in poisonous mushroom Amanita phyloides. This is a very typical poison, and it was used in yesteryears as a death trap or destroying angel. The alpha amanitin is a chemical that binds very tightly to RNA polymerase 2, that because it harms the synthesis of mRNA, but it is rather RNA polymerase 1 is rather insensitive to this toxin, and it blocks the elongation phase of the RNA synthesis. So very vital, it blocks the elongation phase, it binds to RNA polymerase 2, which is responsible for mRNA, but it does not affect all 1 too much, and high concentration of that can block all 3. So this is a comparison of the effect of alpha amanitin, and as a whole we have the comparison of the 3 types of polymerases. This is very important for examination. Very, very important. So this is very, very important. That for examination, eukaryotic RNA polymerases, several times a short note or short answer type question. So this is self-explanatory. We have already discussed. We have to know what are the location of the enzyme, what are the transcribed. Those are the RNA photo, and what is the effect of the amanitin. Please go through that. Next, we come to the most important polymerase, that is the RNA polymerase 2. When I have given it a nickname, the giant. In our younger days, we had a very popular figure in Zamamul Haq, the gentle giant. So the polymerase 2 is a giant enzyme. So the polymerase 2 is isolated from yeast. So why yeast? Yeast is a prototype of the eukaryotic organisms. For the study of the polymerases, we have taken the yeast as a prototype. Scientists have worked on that. So they found it's a huge enzyme with 12 subunits, and there are few important subunits. RBP1 is the largest subunit. Then there are other RBP2 and RBP11. These are the subunits. RBP3. So these are the important subunits of the RNA polymerase 2. So RNA polymerase IBP1 is a similarity with the beta prime unit of the prokaryotic polymerase. RBP2 has a similarity with the beta subunit. On the other hand, RBP3 and 11 has homology to the alpha subunit. So that shows that it has got similarity with the prokaryotic polymerase, and it has got 2 alpha like 2 alpha and beta beta prime. These are the subunits. RBP1, the largest subunit, has a long carboxyl terminal domain consisting of variable number of consensus sequence YSPT SPS. This is important because it's serine residue of these repeat sequences. They may be variable numbers. So it may be 27 number in yeast and 50 in human. The carboxyl terminal of the robust polymerase enzyme, or the carboxyl terminal of the RBP1. So there, the such units are there. The serine residue, they actually undergo phosphorylation, and that can regulate the enzymatic activity. So that is the, but for our knowledge, important thing, if for examination, that's a eukaryotic polymerase, huge, at least 12 subunit are there. Each subunit has got different function, and one of the subunit may be phosphorylated, and it has got some carboxyl terminal domain, may be phosphorylated. The detailing of the different subunits is outside our examination purview. Next, the promoter region in the eukaryote. Once again, I tell you, detailing of this discussion is not in the purview of our examination. But let us see, just for our sake of understanding, some information is needed. So RNA polymerase II promoter, why I am focusing on that? Because it's the commonest. You see that the RNA polymerase II promoter, typically, this is a diagram, there is a Tata box. So just like the Pribnow box in case of the prokaryote, RNA polymerase II promoter region, there may be a Tata box which is an AT-rich sequence. And there may be another type of RNA polymerase II promoter where there may not be any Tata box. So what it happens is, there we see the INR. INR is the initiator sequence. From there, it is actually the start site. So typically in one of the type of RNA polymerase II promoter, there may be Tata box, which is just like the Tata Pribnow box in case of the prokaryotes. But in other type of the eukaryotic promoter, in the type 2, there is an initiator sequence, and the right hand side, that is to the 3 prime, to the plus side, there is a promoter region that is called downstream promoter element. So this is very interesting. We all have so far learned that the promoter sequence should be situated on the left hand side, or that is the minus side, upstream of the DNA, but it is downstream, upstream of the promoter, upstream of the initiation side. The promoter is classically located upstream of the initiation side, but here the promoter is located downstream, and that is why it is called DPE. So this is one important thing, that in case of the RNA pol 2, there may be two type of promoter. One may be in the proper position, that is in the position of the Tata Pribnow box, that is called Tata box, and another may be the DP. And in addition, interesting thing, that both the RNA pol 2, we have got enhancer region. What is the role of enhancers? We may come later. So coming back to the earlier slide, eukaryotic promoters are the cis-acting elements. What is cis-acting? This terminology we may come across several times in our studentship. Cis-acting elements are those elements which are situated on the DNA itself, and trans-acting elements which are situated in other genome and they act on the DNA concerned. Suppose a gene is being transcribed and a DNA sequence is situated maybe on the same DNA but far distance, it will be considered as cis-acting. But a DNA sequence which is located in other chromosome, and then from there some element, some protein is formed, that protein acts on the gene being transcribed. Then in that case, that protein or that DNA is transacting element. So this is the basic concept of cis and transacting. Most commonly seen is the Tata box. So don't confuse, Tata box is found in eukaryotes and Pribnow box is found in eukaryotes. And Tata box generally located in sequence between -30 and 100. But in the Pribnow box, it was -10. It is similar with prokaryotic -10 sequence, but farther from the start side. Important. Tata box is often paired with an INR, which we have shown in that diagram, which is a sequence found in the transcription start site between -3 and +5. This plus this INR is itself a promoter. So one must not forget that Tata is a promoter, INR is a promoter. and binding of the presence of this sequence increases the transcriptional activity. In addition to that, downstream promoter element may be found in those cases where there is no Tata box. It is found in position plus 28 to plus 32. So, with the recapitulation, in some of the eukaryotic transcript, DNA actually we have the Tata box at minus 3200, and we have the initiator element, and we have the enhancer. Enhancer is not promoter. Promoter is this one and this two. And here it is the one, this is two, initiator element, which is around the transcript start site, and downstream promoter element. So these are the promoters. These are enhancer. What is enhancer? We shall come later. Additional regulatory sequences are located between minus 40 to minus 150. These promoters may contain many such promoters. So we are still limited to discuss about promoters. We are not discussing enhancer here. These are the additional regulatory elements. These are found in minus 40 to minus 150. These promoters, they contain a CAT box. CAT is the sequence. That is why it's called CAT and GC box. GC box is more commonly found in constitutive genes. What is constitutive gene? We shall see in regulation. So this CAT box and GC box can be effective when present on the template strand of the DNA. This is very important because CAT box and GC box, they are promoter itself. They are not found always, but they are sometimes found, and they are found in addition to the core promoters. So these are additional promoters, and they are often read at the template strand. Usually, promoter is sequence is read in according to the coding strand, but here it may be the non-coding strand may also, there may be some sequences which act as promoters. So they are include the CAT and GC box, and they are mostly CAT GC is found in constitutive genes. In contrast to eukaryotic minus 10 and 30. So where is the location of the Tata box? Recapitulation. Tata is located minus 30 to 100. And additional regulatory minus 40 to minus 150. In addition to prokaryotic minus 10 and minus 35, TAT, CAT, GC and other regulatory elements are recognized by proteins other than the RNA polymerase itself. So this is very important, that in case of the eukaryotes, in case of the prokaryotes, it is the polymerase which has got the sigma subunit which binds with the promoters, and it is a game end because the binding of the sigma factor, and there is gradual opening of the DNA double helix, and the initiation starts. But here, actually, what is the thing, that it is a RNA polymerase eukaryotic giant enzyme. It often has got its own capacity is very high, but it is not so more agile. Sometimes it needs the help of some other proteins. A big institution has got a big hospital like SSK, so many branches, nephrology, like that. So there may be such thing. So the main enzyme is there, but additional factors are there. So those factors are coming into play, and those factors, what they do, they actually make a liaison. They liaison with the some other proteins, and the liaison of those proteins actually help in the action of the polymerase. So this is a kind of difference, because that is why it is so complicated. So only one enzyme and it being regulated is okay, but there are additional regulatory factors. Those factors will be under the governance of some other factors, and those factors will be synthesized by other DNA. So this is a complete different picture. Now, so this is this picture we have already undergone these basic promoter elements, additional promoter elements, and distally located enhancer elements. So what is enhancer element? These are the regulatory elements. Regulatory elements may be two type: proximally located, these are itself additional promoter, and these are distally located enhancer. So we shall see what is the in the last part, what is enhancer. Now, RNA polymerase 1 promoter. The RNA polymerase ribosomal DNA transcribed by polymerase 1 is arranged in a tandem repeat sequence. Each sequences contain certain copy of ribosomal RNA genes. So as we can see here, 28S, 18S, and 5.8S. So these are the some of the RNA genes, and promoters lie in the region intervening the genes. And there you can see this RNA being the promoter. Ribosomal initiator element and upstream promoter element act as promoter by binding to proteins other than polymerase. So this is clear as daylight. Next, polymerase 3 promoters. What does polymerase 3 transcribe? We have seen they transcribe mainly the 5S ribosomal RNA and tRNA from the earlier slide. 5S ribosomal RNA and tRNA. So two type of the uh, enzyme and two type of the gene being transcribed. So when in case of the 5S ribosomal RNA, this is one type of RNA pol 3 acting here, and this is another type of RNA pol 3 acting here. And here there are two promoter region A and B, A block, C block. They are seen on the downstream to the start site. So this is a typical feature that both these cases it is downstream to the start site, A block, C block. And in case of the type 2 tRNA, that is a tRNA, it is transcribed A block and B block. And it is one is the downstream and it is upstream. But one thing, this is not required at all, it is required in exam. Just remember the enzyme, that is enough. RNA pol 1 and RNA pol 2, this discussion is not required at all for our examination purpose. Now come to the very important, that is the RNA polymerase 2 and polymerase 2, the additional transcription factors. So this is the what I was saying, that the factors, protein factors, which are as a whole required for the eukaryotic transcription. And as you can see, the number one is the polymerase 2. It has got 12 subunits and it has got about molecular weight 10,000 to 2 lakhs, and it catalyzes RNA synthesis. This is okay. It has 12 subunit and each subunit like having different function. But in addition to that RNA polymerase 2, here we have a series of other proteins which are actually synthesized somewhere else, but they are helping the RNA polymerase 2. They are coming into the picture. They are helping the polymerase 2, which is the gentle giant. So this gentle giant needs the help of Tata binding protein. You shall see it has got one subunit and it has got 38,000 molecular weight. This is a very important protein. And binding of the Tata box and the polymerase 2 Tata binding protein binds with the Tata box and it helps in the recognition initiation of the transcription. Then we have the TF2A. TF2 means transcription factor 2, and the specifically transcription factor 2 and the additional letter is A, B, E, F, H. So transcription factor 2 means it helps in the Tata polymerase 2. So it's transcription factor that helps the polymerase 2. And there are several subunits A, B, E, F, H, each having different function. Like A, it has got 3 subunits. It stabilizes the binding of the TF2 and B and Tata box, TF2B and Tata box to the promoter TBP. So TF2B it binds directly to TBP. TF2E binds with the TF2H, and TF2F binds to pol2 and prevents binding of pol2 to non-specific DNA sequences. And TF2H unwinds DNA at promoter, phosphorylates pol2. So these are the different functions of the factors. Some time may be transcription factor, may be a short note, but usually still not important. Elongation factor, there are some other things are there. We shall just remember the important thing that RNA polymerase 2 itself bind to the promoter site, but it cannot function independently. It needs the help of other transcription factors. And the transcription factors acting on with RNA polymerase 2 are the TF2, and series of TF2 are there. They actually formed in different DNA, and they come into picture and they act with RNA pol 2, helping it out to actually do the transcription process. So stage of initiation begins. The Tata box promoters, the key initial event is the recognition of the Tata box by the Tata box binding protein. Tata box binding protein is the TBP, Tata box binding protein. Binding of the Tata box binding protein and Tata box is they are the formation of the TF2D, and TF2D is a complex. and the complex includes other small protein factors also. So we must keep in mind the diagram. When we will refer to the diagram, we shall see the TF2D oftentimes. We should be confused what is TF2D. TF2D is nothing but a complex. To the promoter sequence. And then the Tata box of the DNA binds to the concave surface of the TBP. So TBP is a saddle shaped protein. Saddle shape. So there is a concave and there is a convex. So concave side of the here actually concave side of the DNA, concave side of the protein actually binds to the DNA. So this concave surface, and it is a saddle shape. So the other side of that will bind with the other transcriptional proteins. So other side of the saddle shaped protein will bind with the other transcriptional factors. DNA is caught by the Tata box binding protein, is caught hold of that, and the binding induces large conformational change of the DNA. So it is just holding the DNA and it is inducing a large conformational change in the DNA. Next, formation of the open and closed complex. As in the case of the prokaryote, we have just seen that TBP bound in turn by transcription factor 2B, which binds to DNA on either side of the TBP. Go and see the diagram straight away without any confusion, without much confusion. Here you see the diagram should be much more enlarged if possible, but this is the TBP and Tata box. They bind into the Tata box TBP bound, and we are just showing you the diagram first before any discussion. So it is the TF2B, the large one, TF2B. Uh, So once again, to erase that, and then this is the TF2B. TF2B is a transcription factor. TF2B binds and holds the Tata and TBP on both sides. It actually helps in the anchoring of the TBP. And on the left hand side, it is the TF2A. So TF2A comes recruited and it just supports the Tata TBP being held by the TF2B. So TF2B and A, they are just holding, they are just supporting, scaffolding, holding, supporting the Tata TBP interaction. Thereafter, gradually TF2E and F, they and H gradually come. So this is E, this is H, and this is, so this is E, this is H, and this is F. So what we have to remember that it is the Tata and TBP, they have come and make a arrangement aggregation. This whole complex is being held on both sides by TF2B. This is very important. TF2B. You must not forget the role of TF2B. This is B. From both sides is just like a, it is nursing that complex. And from left hand side, TF2A and has come and caught hold of that, so that the promoter region doesn't slip. But the actual transcription side, start site is somewhat 30 base pairs away, the INR. And there the polymerase will come and do its magic. But let's see and go back to that slide. So the TF2A then binds and stabilizes the TBP DNA complex as we have shown. TF2B provides important link. So TF2B provides important DNA polymerase 2 and TF2B TBP complex is next bind by Pol2 ETF. So Pol2 and transcription 2F. 2F brings the Pol2. 2F is a car which carries the Pol2, and TF2F reduces the affinity of the enzyme to non-promoter DNA. So non-promoter, there is another portion like INR and those portions, those are going to be transcribed. Those portions, the DNA is made little bit weaker by the action of the Pol2F, a TF2F additional factor. And Pol2 has come and now It is the main enzyme, main head cook. Actually, the head cook is brought for the recipe and for the cooking purpose. Now it has come and it has got hold. It has identified where I have to act. Call to F has brought it and left it there. Next come into the picture the TF2E and TF2H. TF2E and H. What is the role of E? As in the earlier diagram, that recruits the TF2H, TPase and helicase activity. And F, TF2F, it binds tightly to Pol2 and binds to TF2B and prevents binding of Pol2 to non-specific DNA sequences. Too much detailing. Most important thing is VP binding with the Tata, recruitment of the factor B holding and A, they make a complex. Promoter region is secured. And other side, TF2F coming and bringing the polymerase to sit over the transcription site properly, and the polymerase and also making a loose association of the non-promoter DNA. Now the third group, third party, that is the TF2E and H, they are actually coming close together. E will bringing the H, and E and H both have got helicase activity, and they will just make the they will act later on. But so far E and H come and form the closed complex. So 2, then 2E, 2F, 2H, 2A, 2B, all these, and the DNA and the TBP, all they form the basal transcription complex or the closed complex. Thereafter, only one thing is left out, that is the TF2H. There are few subunits. They have the DNA helicase activity, promotes unwinding of the DNA near the start site, and this is an ATP dependent process, and automatically it will form an open complex. So previously it was the closed complex, now it is an open complex. This is very interesting, but little bit difficult and may not be so necessary for examination. But additional role of TF2H, we are not going into that. But promoter clearance is important because there is a phosphorylation of the carboxyl terminal domain, as we have said, this of the polymerase, and that thereafter the promoter, after the synthesis of 60 to 70 nucleotides of the DNA, TF2F and H are released, and Pol2 enters the elongation phase of the transcription. So we see the diagram. H and E, they have started their action, and as I have told them earlier, that the H has got helicase activity. This is the closed complex, and now H has made the initiation region, the opened up, unwound, done by the H helicase along with E, because that is easing making a comfort zone for H to work. And H, in addition to its action, starts phosphorylation. So the phosphorylation and the easing, opening up of the DNA and beginning of the transcription, phosphorylation actually helps in the activation of the polymerase, and the transcription goes on very fast. So very quick transcription starts occurring, and gradually the transcription factor E, H, they are released, and also gradually then what happens, elongation factors come into picture and transcription initiation ends. So this is a very tricky thing, but just similar to the prokaryotic one. Elongation, termination and release. TF2AF remain associated with the Pol2 throughout. As I have told, TF2F is suppress pausing during transcription. So they keep the chulha burning, they keep the oven hot, they keep the process going on. They don't allow the pausing anywhere, it should not pause, the elongation factors. Actually, it is the phosphorylated carboxyl terminal domain of the polymerase. If once it becomes dephosphorylated, the RNA polymerase will become inactive. So once the RNA, and there it is helped by the H also. In some cases, H helps in phosphorylation. So anyway, detailing is not required. Once RNA transcript is complete, RNA Pol2 and uh, S is all 2S is dephosphorylated and recycled. So it is ready for another transcript. So in a nutshell, it is the diagram of the transcription process. Transcription all 2. It has got a robust enzyme. It is aided by several factors. They act during initiation, they act during elongation, and so also they act in the termination. There are termination factors. There's a series of factors. And the enzyme itself is a giant enzyme. DNA is a big one. So it is not easy for the enzyme itself to locate the promoter. So to locate the promoter, we have the Tata box binding protein, which actually comes and bind with the Tata box. And thereafter, some of the factors actually stabilize the Tata TBP interaction, and that is there are the TF2B and TF2A, they stabilize. And now the initiator sequence is located somewhat distant from where the actually polymerase will come. Polymerase is already recruited by the TF2F because it is the assistant of the polymerase. But TF2E and H, they come and they help the unwinding of the helix. They have the helicase activity. They also aid in the polymerase to actually not to act in some of the areas where there will be no transcription, non-transcript region. So AF is also helping the polymerase. So AF, E and H actually helping. The role of TF2B and A and data binding protein is now slightly diminished. Now with the help of this AF, E and H, there is unwinding of the helix. The closed complex previously formed is now becoming an open complex. The transcription starts, and right in the junction of the initiation and elongation, there is phosphorylation of the carboxyl terminal domain of the polymerase by in the serine residues by the some of the transcription factors, and some of the few elongation factors also help. And with the factor which bound the promoter, the TBP, then the TF2A and TF2B, they have dissociated, and there is a promoter clearance. Now comes the picture of the elongation factors, and they keep the phosphorylation uh, constantly going on, and the phosphorylation of the CTD, and there is constant polymerase activity, and ultimately there is a transcription, and the end occurs. But we don't go into the detail of the termination in this case. Role of TF2H, it helps in the nucleotide excision repair, the DNA repair we have seen, and genetic loss of TF2H subunits associated with xeroderma pigmentosa and protein syndrome, arrested growth, photosensitivity, and neurological disorders, not required in exam. Now, there are enhancer sequences. Enhancer sequences lie upstream to the promoter region. As we have thousands of base pairs separate the enhancer, thousands of base pairs, although they are cis-acting because they are on the same DNA. They bound by they may be upstream, they may be downstream. Okay. This is the promoter, this is the gene. Enhancer is like situated at a distance. Maybe the promoter is actually the two planets, and enhancer is a star. So it is so distant, thousands of base pairs. But it is in the same DNA, they join because these are the cis-acting usually. So the binding of some factors which are synthesized from other DNA, they are the transacting factors. They bound and they actually bind to the enhancer, and they sometimes the enhancer then will act with some of the proteins, and the binding of the it will influence the polymerase activity. The activity of many promoters are greatly increased by another cis-acting elements. Those are called enhancers. Although do not have promoter activity, they can exert stimulatory functions over distances of several thousands. They can be upstream, downstream, or even midst of a transcription. Enhancer sequences are acting. Particular enhancer is often effective only in certain cells. Immunoglobulin enhancer effective only in B cells. So study on enhancers is a very tricky subject, and it is a very upcoming subject. Several, as I have said, mentioned, enhancers act through formation of protein factors. Several protein factors. Actually, what we have described here is a basal transcription complex, but there are thousands of protein factors. They actually can regulate the action of these basic transcription factors, and the synthesis of the those protein factors may be under the regulation of enhancers, and the enhancer DNA can directly also act on the some of the protein factors. So this is a very complicated and upcoming subject, and overall the regulation of transcription is a area under the regulation of gene expression, which is uh, the latest on the molecular biology, because all the new drugs are being synthesized are based on the concept of this regulation of gene expression, how a particular gene can be knocked out or how a particular gene can be expressed more in number. So this is a completely different subject. But so far, with this much of discussion, this much of talk, I think we have done an exhaustive discussion on the subject of transcription. Thank you all.